Hello, I am Marco Ruggiero, although the voice you hear is not my own, but borrowed from AI for reasons of clarity. As some may recall, we have been present at major international conferences on autism since 2014, and today I would like to provide an update on the scientific results concerning the relationship between the microbiome, its alterations, and the autism spectrum. Recent observational studies confirm distinctive microbiota alterations in children with autism, characterized by a reduced abundance of bifidobacterium and an increase in Clostridia and Enterobacteriaceae, which are correlated with the severity of social and sensory symptoms. The proposed mechanisms include the altered production of short-chain fatty acids, dysregulation of serotonergic and GABAergic pathways, and low-grade systemic inflammation that influences white matter development and neural connectivity. Microbial metabolites, such as propionate and indole-3 propionic acid, modulate microglial activation and myelin, creating a biological bridge between dysbiosis and brain alterations. Therapeutic interventions are showing promising results. For example, a 2024 multi-center double-blind trial reported significant reductions in behavioral scores and improvements in gastrointestinal symptoms after 12 weeks of probiotic treatment. These findings confirm that the microbiome acts as a key metabolic organ capable of influencing brain function, suggesting that the restoration of microbial biodiversity represents a fundamental therapeutic frontier for the management of neurodevelopmental disorders. If you are interested in learning more about our research and the Bravo microbiome reconstitution system, visit our websites or contact us via email at info@bravoeurope.com or through our social media.